Hi there. In this video, we will do an end-to-end analysis of an ATAC-seq dataset on Pluto, going from raw FASTQ file upload all the way to publication-ready visualizations in a matter of minutes. For this experiment, we're going to use ATAC-seq data taken from zebrafish at 3 different developmental time points, treated with and without retinoic acid. So you'll see here, this is really kind of a completed analysis on Pluto. You'll see publication-ready visualizations like differential peak analysis, gene set enrichment, IGV plots, and then you'll also notice that there are text boxes where we can really summarize the findings of that data. And this is really intended to help you tell a cohesive scientific story to prepare for publication. What's important to note about all of these visualizations is that they are interactive and you can change the colors, change labels, change text and dimensions, and really get them into that publication-ready format. But let's start at the beginning with our FASTQ file upload and talk about how exactly we got here. So to initiate an experiment on Pluto, you can select an experiment type, in this case ATAC-seq, as well as an organism. Then you can add your assay data, and you can either start with FASTQ files and run an alignment and quantification pipeline, or upload a CSV file of consensus peak counts. Then you can upload your sample metadata and configure your pipeline to run. So this will run NF-core ATAC-seq, which is generally recognized as a gold standard pipeline. It's very easy to view all the code for that, as well as cite it in your method section. Then you'll just select a few parameters like genome and library type. And then by clicking Run Pipeline, that will kick off the bioinformatics pipeline, and it will also initiate this experiment view. So you'll see the notebook, which is where you can input different information about the experiment, like the hypothesis and the experimental design, and really take notes as you go. This is also a great place for experimental planning. This can be created before you even have data. And then down below, you'll see all of the data associated with this experiment. So you can see the sample metadata, as well as the assay data. So this is the consensus peak counts that came out of the NF-core ATAC-seq pipeline that was run. And then you'll see the pipeline QC report. So this contains information like the max2 peak count, as well as the sequencing quality. Then you can start your analyses. So let's head back over to the canvas and kick off a new analysis. So you'll select Create New Analysis from this plotting function here. And then you'll be able to see all of the different analysis modules that are available for this data type. And you'll see that most of these analyses come with more than one different plot type that can be created. So let's go ahead and select differential binding and click on Create Analysis. And then you'll be able to create experimental groups from the different metadata variables that you uploaded when you initiated your experiment. And you can actually create groups combinatorially, so you could just select one variable to group by, or you could select developmental stage and treatment to create smaller groups. So you can select 12 hours retinoic acid as your experimental group, 24 hours, and then 24 hours DMSO as the control, and then click Run Analysis. And that will kick off a bioinformatics script in the cloud that typically takes 10 seconds to a minute to run, and then it will deliver a nice completed volcano plot. From here, you can edit the volcano plot to meet your specifications. So for example, you could add in p-value threshold line and log-fold change threshold lines. And you could also change the colors of each of these kind of individual points. So let's go in and just change the significantly increased peaks to be pink. And then perhaps most importantly, there are a few different ways that you can call out genes of interest. So you could individually label these on the plot just by clicking on them, or you could create an entire list of genes that you're interested in following and select those from this biomarker target list dropdown, and that will label all of the genes together. Every plot that is generated on Pluto comes with a method section that auto-updates based on the parameters that you set when you configure your analysis. But when you complete your experiment and you have all of your visualizations ready to go, you can also create an entire method section that fills out the methods for every single plot that you generated. So when you're done with your analysis, you get all of your publication-ready figures, your method section, all of the text that you have generated to summarize these individual plots. And then this entire workspace is also collaborative. So you and your team can work towards creating this cohesive scientific story together. And an entire analysis like this can really be done in 45 minutes to an hour. If you have ATAC-seq data and you're interested in seeing it on Pluto, we'd love to talk to you, so feel free to email us. Go to pluto.bio. Thanks for watching. Bye.